I study, and my lab studies how the brain develops a little bit differently than the early minds lab, which I think is a huge strength of our program. We can ask interesting questions in a lot of different ways at a lot of different levels. So what I'm interested in is development of sex differences in brain and behavior. So specifically, a question that's always kind of fascinated me is as mammals, we come together, egg, sperm, moment of conception, one cell. Everybody in this room started as one cell. And then a few weeks to months later, depending on your species, we have a pretty good looking brain at birth, pretty functional, does what it needs to do. That's a massive construction project, right? To go from one cell to this functioning brain. That lots has to happen correctly. And at that moment of conception, we already have a difference, XX individuals versus XY individuals. And so I'm interested in how does this massively different or this massive construction project work differently, if at all, in XX versus XY individuals? I want to make sure I say I work with a rodent model. I'm interested in mammalian brains. So I'm going to be talking about sex differences, not gender differences. It's a different question. So, all right, so what could be different between males and females within this process, and what are the consequences of that process? Are there sex differences after this process? And if we look in humans, one of the biggest behavioral differences that we see is this observation that boys have a higher incidence of developmental disorders like autism spectrum disorder. It's about 4 to 1, so about for every 4 boys that are diagnosed, there's one girl. Some, depending on how you define it and look at the symptomology, it can be up to 9 to 1. And it's not just ASD. Think about some sort of psychiatric, neurological disorder, anything that comes to mind. If it presents in childhood or early teenage years, it's diagnosed more often in boys. Pretty much. I can't say universally, I'm allergic to that as a scientist, but almost always. Most disorders that present in adulthood are seen in women more than men, although it's a little less profound of a difference. So if it's all disorders that we can think of, if it's autism, if it's Tourette's, if it's early onset schizophrenia, if it's stuttering, if it's ADHD, that means that there has to be something different between male and female brains. It has to be the result of this process because it happens pretty early in childhood. And we're kind of left with a conundrum of, okay, it's this huge difference. Well, that means it can't be like a brain area, right? It can't just be like, oh, it must be the hippocampus that's different, or it's this gene that's different. It has to be something that's really global. Okay. Well, it also has to be something that's pretty subtle, right? Because it's not like every boy gets a disorder and no girls get disorders. And it's not like everybody gets the disorder in the same way. So what could it be? I and a lot of others have started thinking about this. What is the mechanism that we go from conception, we go from XXXY, we go from differential hormone exposure, spoiler alert, to a lot of behavioral and other molecular and cellular differences. What's in the middle here? And one of the things I've been thinking about, along with a lot of other people, that could be both global and subtle, is that we think about how neurons communicate. That's their job. Their whole job in life is to communicate with other things, mostly other neurons. And they do that through electrical means, excitation, inhibition. So one idea that can be fit this global but subtle thing is that turns out male brains, we have some data, lots of other data out there. Male brains, male neurons in general, tend to be more excitable. I think I keep doing this because I think of it as sort of like a dimmer switch on a lamp, like it's sort of tuned to be more excitable. Female neurons, on average, tend to be a little calmer, a little more stable, for lack of a better word. This could lead to all kinds of differences depending on exactly how far the tuning is different, where it's different, and when that difference shows up. So that's that black box. What is actually happening within a neuron? What is actually happening within the cells, within the molecules that's leading to these excitation inhibition changes, and then ultimately these behavioral outcomes that we see. Does that make sense? So one of the things we're working on in the lab is this structure called a perineuronal net. This is kind of a newer thing. You may not have heard of it, PNNs. They're kind of what they sound like. They're net-like structures. They are around neurons, specifically around synapses. And what they do is they're extra cellular matrix proteins, and they kind of start to assemble themselves across late development, early postnatal life. And at some point, they kind of crystallize and kind of lock down that synapse that they're surrounding. Turns out that that locking down corresponds with the end of critical periods. And we know other folks have done experiments where if you dissolve those perineuronal nets, you can actually reinstate sort of a level of plasticity in adulthood that you haven't seen since the end of the critical period. As per usual, no one had looked at a sex difference, though. It was all done in males or unknown sex. And so a couple years ago, we published that this was a sexually dimorphic. This was at the same age in the hippocampus. These little green things are the perineural nets, the red things are the neurons. And at this particular age, in males versus females, we have them sort of starting to crystallize already, not so much in females. So now we're trying to figure out what that means. Like, what's the downstream effect of that? In general, types of things that I use in the lab, because we're interested in that cellular neuronal sort of functioning, we use a lot of molecular techniques like immunocytochemistry. Let's see, we do behavioral assays to see what the outcomes are. Do we have an effect on behavior? And one of my favorite techniques that we have in the lab right now are sex-specific cell cultures. So I don't know, maybe you resonate with this or not, but I think all scientists at heart are control freaks. One reason why I chose rats and not humans. Turns out you can get even deeper level of control. You can take the neurons out of the rat, put them on a dish, and now I have a dish of male neurons and a dish of female neurons, and I can do whatever I want to them. I can poke them, I can throw drugs on them, I can throw hormones on them, and I can see how they respond. So I can basically ask the neurons direct questions. And this is partially how we got to this excitation inhibition question. Turns out if you throw on glutamate to both male and female neurons, on average, male neurons sort of pop off. They fire a lot of action potentials, and then they die. Female neurons don't seem to sort of fire as much, and they don't die as much. That is part of the dissertation of my graduate student right now, Ellie. So she was in here before. You'll see her again this afternoon. And just some other things that we've looked at, because in general, if it's under this umbrella of sex differences and how they develop and the outcomes of that development, we look at it. So some of the things we've done over the years was, excuse me, to look at seizure behavior. So if you think about excitation inhibition, the extreme of excitation is a seizure. And so it turns out, yeah, the way seizures sort of happen and the way we can control them is different in males and females. We have a model of autism spectrum disorder called developmental hyperserotonemia. And it turns out that, again, there's a sex difference in sort of how that model plays out, how the brain looks, and how behavior happens. In this case, peer-directed play behavior in juveniles. And then here's our perineuronal net publication that we've also been working on. So I think I'm probably out of time. Like I said, I have to run, but if there's any questions, please flag me this afternoon or email. Have a great day.